So, welcome back to this course on functional genomics. If you recall that we discussed human genome project when it was initiated in 1990, it was majorly funded by the US government. Not many other countries could join because of the cost involved in sequencing. And also the infrastructure required and all the technology required. So, you can see that if you look into the the capability kilobases per day per machine kind of calculation if you do, you can see that in 1990 when people are using manual slab gel to automated slab gel to what do you have capillary sequencing, it has, you can see that it has really increased the speed at which we are able to sequence. But with what is called as massively parallel sequencing or next generation sequencing, the speed at which you are able to sequence the DNA has tremendously increased. Now we are looking at almost probably in the future is single molecule, just isolate one DNA and then look at what are this transcript that are made. So one can do and go and look into that. At the same time, there is another important element that really helped us is the cost per genome has dramatically fallen. Now you can see that when the genome sequence was at peak, when the draft sequence was about to be published, you can see that cost per genome, for example human genome, is about, and you can see that it is $100 million to sequence one human genome. But now it has come almost like $1,000 you give, you will be able to sequence the human genome. So that is one of the greatest benefits of the projects, because now one has to sequence the genome to understand what changes you have that may give you risk or of developing some disease. Then it is doable because it is $1,000. Maybe with time it can come down to a few thousand rupees. So that is a tremendous achievement. So that's something which is unexpected and which did not even follow what is called as Moore's law, which this Intel co-founder said that this kind of technology is exponential growth, but this is beyond that exponential growth. It is really even beaten that law. So that's the power of the what is called as next generation sequencing. Let's see what is the difference between the Sanger sequencing that we discussed and the next generation sequencing that we are talking about. So the first thing is the strategy. So if you see that Sanger sequencing, you have to separate reaction for sequencing of all exons of a single gene. So you have to, for example, individual exons should be taken out and put in a tube to sequence the small segment of the genome. So here in the next generation sequencing, one reaction for the entire genome, as many segments can be sequenced. That's the huge difference. So you don't really do independent reactions. So what is the use, major use of Sanger sequencing? So if you are looking at a particular segment of the gene, then identification of unknown mutation by sequencing, that is the use. Then this next generation gives you much better way, that is, for example, analysis of unidentified mutation heterogeneous disease, right? You don't know what it is, but you really want to look at everything, so you will be able to get some changes that possibly lead to the disorder. The benefit is Sanger sequencing is still better in terms of precision, because the accuracy with which it will be able to tell you a change in the DNA is very high. But whereas in next generation sequencing it is highly cost effective, you don't spend that much money, but it's efficient because it is very fast, you can finish it very quickly. That's a huge benefit. What is the disadvantage of Sanger? It is expensive and takes a lot of time. And then limited automation, many are still requires manual approach, whereas in next generation sequencing the interpretation of the data, that becomes challenging because it really requires skilled labor, because it produces huge amount of data and at times the accuracy may not be that high. So high coverage needed for accuracy. So when I say accuracy, high coverage, a small segment, if you want to understand what changes are there, that needs to be sequenced at least 50 times. So that's where you come in accuracy. Let's see how it is done. So this is the Sanger method. So, basically we described already that these days people use PCR fragments. You have primers with all the 4 bases with one of the dideoxy base, and then you separate in a capillary, and then you read out the sample depending on which fluorescent moiety is being released, right, at the end of the capillary, what comes out, what fluorescence it gives. That is how you read the sequence. Whereas in next generation sequencing, so what you do is you take the old DNA and then you fragment them. So, that is what is shown here. So, here you have a single fragment, so that is multiple copies of that particular region of the DNA. Here you have fragments of certain range as desired for a given chemistry, but these are heterogeneous. They do not represent a particular genome. It represent the entire genome if you are looking at the entire genome. So, what you do is that then you have what is called as adapters, so which are added at either end of each one of the fragments, right, and then to create what is called as a library. So, here every fragment that is generated from a given species in terms of DNA is ligated, joined with the adapters that are shown here. So, these are identical sequence added on either side of the DNA to make what is called as a library. Now, these libraries are used for what is called as cluster generation. So, basically what you do is that you have a microfluidics kind of a chamber in which the individual DNA molecule get in, and then because it has got the adapters on either side, you have regions that are complementary to that. One or few of the molecules will make a complementary sequence, and then this you do a very similar kind of approach where like DNA polymerase is given, so it makes multiple copies that we will see little later. Once you make multiple clusters, meaning each cluster now represent several copies of the identical sequence. It is exactly like what is used here, but everything is done by automation mode here. So, this cluster is having identical copies, right, and then you are going to sequence each cluster and then read the sequence. Then now that gives you the sequence of all the fragment that you are able to capture in this method. So, that in one go you are going to look into large number of samples. So, the question is how do you really develop these clusters? There are different methods. We will see one of them. So, basically what you have is you have some solid devices in which you have these sequences that are complementary to the fragments that are having these adapters. So, the sequences are complementary to the adapters. So, when you send these fragments that are having the complementary adapters into the devices, the fluidic devices, then they will go and anneal to these sequences because of hydrogen bonding, and then people add, for example, all the 4 bases and DNA polymerase, then it would extend, it will make copies of it, and then you are able to create clusters like that, which I will be able to explain in a movie that I am going to show the next. So, once you make these clusters, right, so as I told you, these clusters represent DNA copies. This is almost like a cloning, so it is like a PCR. So, one copy, now you have several copies of the same sequences, and they are going to sequence over there. So, the way you sequence, it differs depending on which platform you are using. So, the one is sequencing by synthesis, the other one is pyrosequencing, and the third one is sequencing by ligation. So, there are the 3 major methods people use in next generation sequencing. In these first two methods, what you basically use is a DNA polymerase. Obviously, you have a primer, you have a DNA polymerase that goes and extend, and there are bases that are having different modification for scoring whether it is which base it is. Whereas sequence by ligation does not involve any DNA polymerase. Let us look into these two. So, in the first one, the sequence by synthesis, what happens is that you have a primer that binds to, these primers are nothing but exactly the same, adapter sequence that is present in all the DNA of the library. So, once it goes and binds, then what you do is the different between this and Sanger method is here the bases that are used for calling the base, whether it is A, T, G or C, the way it is done is very different in this chemistry. So, you have a base which has complementary, say for example, this is the first base after the primer, the polymerase has to find a base that is complementary to T, so obviously it will go for A, and when it goes, this base is all invariably having the terminator, right, there is a terminator cap. So, at a time only one base can be added. So, once it is added and then you all the reaction in a cluster will add one base and it will stop, okay, and then you wash out all the bases now. So, no other base is left, and then you excite them with a laser and then see what fluorescence it comes out. If it is A, it is going to give a particular fluorochrome, and therefore, the machine is able to read it as A. Once the reading is done, what you do is you basically remove the terminator, like what is fluorescence taken. After that, you remove the terminator, you also remove the fluorescence moiety from the base, then you wash it, again you give all the 4 bases with the terminator and the fluorescence, and then allow the reaction to go and add another base, again read it, again cleave it, again wash it, again go for the third basis. So, this is the way you do it. So, in a cluster you will find that all of them, all the molecules that are present in a cluster that we are shown here have the identical sequence, therefore, from the base they will add one base. So, all of them will be excited for a given fluorochrome, therefore, you are going to get the fluorescence like this. So, your machine would call it as A for the given dot. So, this would be A, this would be B, C or whatever it is, and then you go on doing the reaction and you are able to complete it. In pyrosequencing, it is depending on the reaction, a kind of a chemical reaction that happens here. So, you have a reaction in which whatever moiety that are added, the the chemical that is released would give different color depending on the base, right. Again, it is a similar method. What fluorescence comes out for a given base, that would be read out. The difference is, at a given time you are going to give only one particular base. For example, you have the primer that is annealed to the template. So, then you flush the system with only the base A and then see whether that got added. If it got added and when you do a reaction you will get a fluorescence. Suppose that is not A, then this will not get incorporated, so you will not get any fluorescence. So, it could be G, so you will get a fluorescence. So, you for everything you add one base and see whether fluorescence comes, wash it out, give the other base, wash it out, give the fourth base. So, in this way you can identify at the same time whether a base is added or not added, accordingly you can call the sequence. So, this is what is done in this case. So, this is the way they are able to determine the sequence. The ligation method is very different. You do not use any DNA polymerase. Here you have oligonucleotides that have combination of various bases. The only difference is that the first two bases are fixed. So, you have either, for example, AT, CT, TT, GT, for example, these are the bases like what is shown here. This is the first base, this is the second base. The rest of the bases could be random, all combination exist. So, now it all depends on whether, for example, you have a primer that is aligned to your sequence which is single stranded. Now, since the primer ends here, you have to have a small oligonucleotide that is complementary to the first two nucleotides of your template. If it is GA, so you should have the CT containing oligonucleotide that can come and anneal. Once it anneals, the ligation reaction happens and this is sealed to make a larger fragment, right. So, then the system is able, because this is annealed and this gives you a different fluorochrome, therefore, the system is able to call it as that this next two sequence after the primer is C and T. Then you have another reaction that goes on, you remove this fluorochrome, then you have another base that comes in, and again this would be calling for whatever bases that is present after the first oligonucleotide is annealed. So, now in between you are not able to read, but after 3 base you are able to again read the 2 bases of the DNA and go on, it goes like this. And then you have different primers that anneal at different segment. For example, you can, this is the primer, if you consider that ends at 0, then it anneals at 1. You could have other sequences annealing at -1 or -2 or +1 or +2. So, therefore, you are able to read the entire segments and get the sequence. So this is another method which is not very successful, but these are the two most common chemistry people use for sequencing using next generation. So we are going to have a video that is playing to you, movie that is come from the YouTube that I am going to show you now, and this particular website you can go later as well and then watch the movie.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2. Each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequence of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing, is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The iron proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com, and from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service.
So this is just to give you an overview that you have the DNA that is cut and made into fragments, and you have the adapters that are added on either side, and then flow them through the grids or the chambers that are present in this cell, and they all go and bind depending on what the adapters complementary sequence present over there, and then you amplify, make clusters of identical copies, and then you have the DNA polymerase adding these bases and the image comes and you are reading. So that's what's going to be shown in this video.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next generation sequencing.
So what has been shown is the Illumina platform, what is called as Solexa sequencing principle, and this pretty much gives you a comprehensive sequencing coverage of the DNA. So this DNA what you are using could be any DNA, it could be the genomic DNA, it could be the cDNA, meaning you have made the copied DNA of the mRNA, now you are sequencing the cDNA, therefore it represents the mRNA. Or it could be the chip data, meaning the DNA that are bound to certain transcription factor or histone and so on. So any DNA can be used here to sequence, and that would tell you what is that sequence that particular DNA represents. So that, this is a versatile platform, you can use it for any, and some of these usage is shown here. One of course is the whole genome sequencing, so you can sequence the entire genome using this method. Or it could be what is called as whole exome sequencing, meaning you do not sequence the entire genome, you are looking at any disease, you are likely that the disease is caused by a defect in the coding sequence. So what you do is you pretty much select the regions that code for the genes, right, the protein coding sequences. Therefore you selectively enrich these regions and then sequence the whole genome. So it depends on how do you capture this DNA. Or you can restrict your analysis to a few set of genes, not necessarily the entire gene, but the regions that are most likely mutated in many of the family, for example clinical applications. So you can go and look at point mutations, deletions and so on. So you amplify small segments and go and look at a large number of genes but restricted to small regions. Or you can start with the RNA and convert them into cDNA and use the cDNA for understanding gene expression, because this also not only sequences the RNA, but it gives a quantitation, meaning a gene is expressed highly in a given tissue. So you are likely to have more cDNA derived from that RNA because you have more copies of the RNA. And if you have read many of such sequences, then the reads, number of reads for a given gene would tell you the expression value itself, because it is not only qualitative, but it also gives the quantity, because number of reads represent number of copies which represent the expression level of the gene. And you can also understand what is called as gene expression with regard to, for example, the splice variant. Remember we are discussing how the gene can undergo alternative splicing. So most of the microarray you have a limitation with regard to what genes, which splice variant you are looking at. But since you are sequencing here, and if you restrict your sequence analysis to exon, intron or exon boundaries, you will be able to tell which spliced form is more abundant, which is less, right. That really helps us. So that is another big advantage of next generation sequencing. So what you are going to look at is some of the examples of how the next generation sequencing has helped. Here is a paper which talks about development and validation of a whole exome, meaning here the sequencing is restricted to the exome. Sequencing test for simultaneous detection of point mutations, insertion, deletions and copy number alterations in precision cancer care. So you really want to type the cancer and tailor the therapeutics based on the cancer type, and you have to do it quick because the patient is already in the hospital. So you removed a small cancerous region and then you extracted the RNA and the DNA and go and sequence it. Within a day or two you are able to get the analysis, which tells you what kind of changes happened, which is not restricted to point mutation where one base changed, but it is also involving what is called insertion deletions. There are regions in the genome, especially in the intronic region or in the promoter region, where there are repeat elements that are present either in 2 copies or 1 copy. It is called the insertion and deletion polymorphism that modulate the level of the expression or the splicing or the functioning of the gene. So you can correlate that, and then it is also called as copy number alterations. There are segments in the genome which undergo multiple copy number variation. They get amplified in cancer. The number of copies go up. So this, since you are unlike Sanger sequencing where you are cloning and sequencing, which do not tell about the quantity or the quantitative measurement. This next generation sequencing, it talks about how many reads, how many molecules you are able to read. It also tell you the copy number variation. That is a huge advantage of this next generation sequencing. We are talking about such kind of panel, one that came published recently. It talks about the frequencies of somatic mutation in cancer patient, because it's a huge challenge. As you know, the cancer is not just one gene that is mutated. It is a large number of genes are mutated as a result of cancer. The number of genes that are mutated could vary from one region of the body to the other, even if it is the cancer is undergoing metastasis, or depending on the cell type. So this is one such analysis where they have used the next generation whole genome sequencing to understand what are the changes that are present here, right? On different cancers, that is, say these are somatic mutations per megabase pairs. You can see that it varies from patient to patient, right? Depending on what kind of cancer you are seeing. That really talks about the heterogeneity and the need for such kind of approach, because information that you get from one patient is not going to really help the other patient. So you need to understand what is so unique about that particular cancerous population found in a particular patient. That is the tremendous advantage, and understanding such kind of complexity involved in cancer is also, now you have what is called as Cancer Genome Atlas, which is kind of a repository which adds all those data into the database. So one can go and look at and then come up with a better way to analyze the cancer. With that, we will end this lecture. In the next lecture, we will see a few more applications of the RNA-seq or high throughput DNA sequencing technologies.